The Conversational Flow, a KCAS podcast.
Hello, and welcome to the 16th episode of The Conversational Flow, an official podcast of KCAS Bioanalytical and Biomarker Services. KCAS is a bioanalytical CRO serving the pharmaceutical and biopharmaceutical industries for over 40 years. Unlike KCAS' other podcasts, the Weekly Bioanalysis, which features news and topics more generally associated with the world of bioanalysis, the Conversational Flow focuses more on the niche of flow cytometry and its role within the greater industry. My name is Adam, and I've been in the flow cytometry space for over 15 years. I'm here with my co-host, Brian.
Thanks, Adam. My name is Brian. I've been in the flow cytometry CRO space for about 13 years.
We also have special guests with us today, Allie and Ayman. Would you like to each give a brief intro?
Sure. Thanks for having us today. My name is Allie Greenplate. I am the Director of Strategic Alliance for the Institute for Immunology and Immune Health at the University of Pennsylvania. I got my start in flow cytometry back in the Johnson & Johnson days on a Canto, and have since gotten a PhD in immunology and moved on to the cytophs, spectral cytometers, and other new flow cytometry techniques.
Hi, my name is Ayman Rezk. I'm the flow cytometry lead at Immune Health. I've been in the field of immunology for well over a decade, having worked previously in the field of neuroimmunology. And yeah.
Excellent. Thank you both. Looking forward to talking with you on our podcast today. Brian and I are members of the growing leadership and scientific advisory team at KCAS. Either or both of us are available to answer any questions you may have regarding this podcast or any of KCAS services. While our KCAS headquarters are located in Kansas with additional locations in Europe and Australia, we're both here in Philadelphia, and Jeremy, our producer, is in Missouri. So we're truly all over the map. We're excited to have you listening to our 16th episode of our podcast, which will be available virtually everywhere. Wherever you choose to find and play your podcast, you will likely find the Conversational Flow. We welcome all of you, whether you're joining us for the first time or if you're a regular listener to any of our other podcasts. Today's episode will be a review of the latest news and resources and then a focus on a topic of our choosing before discussing any feedback we receive from you. We're constantly looking for topics, and we'd be happy to discuss something that you want us to cover, so please reach out. Again, we're fired up to have you here and looking forward to a fun episode today. To kick us off for episode 16, Brian is going to go over news and resources before diving into today's topic. Brian?
Thanks, Adam. We've got an exciting agenda today, and this is going to be a little bit different than some of our other episodes where it's entirely focused on one, I'll call it indication, but it's really sort of a group of indications. So all of the things that we'll talk about today are related to autoimmune or immunology research. And the first sort of medical research news item that we've got today is some additional information that we got from Sana Biotechnology on their cell therapy for type 1 diabetes.
So can I take an aside? Did you ever watch this cartoon, Pinky and the Brain?
I did.
You did. So you know what they said. What are we going to do today?
Same thing we do every night, Pinky.
Same thing we do every time. You said we have an exciting agenda, and I think it's exciting every time. So I just, for some reason, my thought process went there. And I will say, I have to clarify, my thought process has like 5 trains all over the place. And so I just had to digress there and all on that line. Yes. So getting back to the news here, this was really exciting for me because of a couple of different points. One specifically is the type of therapy that we're talking about in allogeneic therapy, right? Cell therapy that is off the shelf. And it's a big step because a lot of these cell therapies are autologous and require taking the cells from the patient themselves, modifying them and putting them back. Whereas this sort of therapy is built and can be taken off the shelf and given to many different patients, right? And it's really kind of what we've been looking towards is almost the holy grail for cell therapy in that respect. The other thing that was interesting too is the fact that much like I think 2024 happened with many companies that we know, they shifted to autoimmune This was a company that was focused in large part on oncology, which, I mean, honestly, CAR therapy had really cut its teeth on oncology and now is being repurposed. And so looking at this, I was most interested in how do they get around, rejection, right? And so for cell therapy, that's the biggest thing when you're looking at allogeneic. And so, curious, Brian, what were your thoughts here?
Exactly, and that was the other really exciting thing. I think that's why the stock basically responded the way that it did to this announcement. The fact that they were able to use allogeneic, which just significantly decreases the cost of this treatment off the bat, but then B, they don't need an immunosuppression regime to
Right.
do so. That's wild, and that's I think the enabling technology or the enabling leap to move some of these cell therapies into the autoimmune space. For oncology, it's it's an acceptable risk and an acceptable cost to need to suppress the immune system for some period of time and to do things like autologous therapy where it can be extremely expensive per treatment, to the tune of between $500,000 and $1 million in some cases for a single treatment. But it's a life-saving therapy, so it's medically and financially potentially feasible. For things like diabetes or other sort of less severe autoimmune diseases, there's curative regimes that are possible. And yes, there's always a financial impact. Like no one wants to spend $500,000 so they don't have to prick their finger anymore. But there's also the risk aspect, and if you have to do the immunosuppression regime, that's a big enough risk that I feel like a lot of doctors and patients would consider that too high of a barrier to get the general public on it. But without that immunosuppression required, then that opens up a whole new level of possibility for treatment of autoimmune disorders, and specifically diabetes, in this case with cell therapies.
All right, and that kind of leads into the paper that we'll be doing today. And immunosuppression can lead to infection, right? Your body has an immune system for a reason, but you also want to prevent it from destroying the therapy in this case. And so this was exciting. It was a single patient. And it's 4 weeks post-transplant that they saw that it remained, and they were so it seemed like it's encouraging, but certainly more to come. Modifications, as you might expect, modifying the HLA-1 and 2 to avoid the adaptive immune response. But then what was interesting is a lot of times you do that, you still have the innate response that will kick in. And so they've overexpressed CD47. Now, CD47 is ubiquitously expressed, meaning it's all cell types. However, they overexpressed it, and then that essentially acts as a don't eat me signal, for example. So you're not engaging your MACs and specifically your NKs as well. So just really cool to see how different companies handle this. I did have a question for you. I meant to look this up beforehand. Sometimes I see it as allogeneic, and other times I see it as
Allogenic.
And so is there a difference?
I don't think so. I always thought it was like the aluminium thing, right? Where it's just different accents. What's your take on that?
Yeah, I've only seen it as interchangeable, so I kind of assumed similar, but I didn't know if that was just me not diving into it.
Okay, we will open it to a wider audience later and get other people's takes on it. But that's a good point. Mm-hmm. But I don't want to say the whole time on that, although we probably could. But it is definitely an exciting development, and right along in there with autoimmunity and immune therapy. People sometimes forget that some things like diabetes and rheumatoid arthritis are definitely in the immune space because they've just been there forever, and everybody's looking towards new diseases like our next story on lupus. But indeed, those are huge sectors of the market that are, ripe for disruption there as well if the right treatment comes along.
For sure.
So with that being said, moving on to our next piece of medical news, this is just highlighting some of the different things going on in lupus research. And for that, I directed us to a blog put out by the Lupus Foundation of America.
Now, the thing that struck me here, I've always been aware of lupus, and many people picture it as having the butterfly rash, the malar rash that occurs on the face in 30 to 60% of people with lupus. But one of the things I didn't realize was really a definition for remission. So there was a task force that was put together called the DORIS, DORIS task force, and essentially identified what does remission look like. And so mentioned that the disease activity index would be 0, physician assessment of 0.5 or lower on a scale of 0 to 3, and then prednisone usage of no more than 5 milligrams daily. It's important because I think my understanding of lupus is it's very heterogeneous as far as there are different types, and the treatments aren't all universally effective. And so what they talked about here were small molecule treatments, antibody treatments, and then again, chimeric antigen receptor treatments. There's even an allogeneic anti-CD19 CAR NK.
That's it, right? And you've hit the nail on the head where lupus itself is potentially sort of a constellation of different either diseases or disease presentations that is impacting people differently. Some of that is our own ability to specify what's going on in the immune system, so understanding what the specific mechanism is. But you're absolutely right that a common measuring stick, it seems basic, but it can be very difficult to develop. And it's actually one of the big things that holds a lot of different diseases back. This is very similar to Alzheimer's in a lot of respects, or some of the other neurological diseases, where if we're not sure if everything is the same, then it can be very difficult to try to treat anything, because anything that you do will have differential impacts. And if you just happen to be testing on, let's say there are actually 3 different diseases that make up what we think is one disease, right? And if you're testing and your patient population happens to only have subset A and B, and your treatment's only available on C, you're missing out on a lot of therapeutic benefit that could be garnered if you could specify only subgroup C. So it's absolutely imperative that we have these types of benchmarks and measurements, and absolutely imperative that we keep up this level of sort of multiple approaches as we go through some of these complex diseases like lupus.
Right, and to your point, and again referencing the paper that we're going to be talking about, they had mentioned that one of the monoclonal antibody therapies is used as an off-label for lupus because it doesn't broadly apply to the entire population, but it does show some improvement over control, and so over placebo, and so essentially there's something there, but it's again defining that population. And it's even just how you define if somebody has, or determine that somebody has lupus, involves several tests and symptom review. So very challenging.
Right. But a lot of progress being made, and that's what we're very excited about, both with this podcast, but everything we do at KCAS, and with some of our guests that we'll introduce in just a little bit. Mm-hmm. To get there, we'll sort of run through our investment news as well. So the first one is just a look back, which is sort of an easy target for the January episode of our podcast here, to basically look back at 2024 and evaluate what got funded and what didn't.
With this, I was interested to see the top ones. They mentioned some that were $1 billion worth of financing. Hopefully I pronounce it right. Zyra, was number one there in over $1 billion in financing. It's an AI-based company looking towards therapies. And I was interested too because the board includes Alex Gorski, who many may know was the former head of J&J, and I bring that up just because having worked there before was cool to see. But they really do have a lot of called star power on that board over there, and so I think that's one of the reasons why it really attracted, between that and the AI, of course, the technology, what attracted all that investment. Second on there was Treeline Biosciences and looking at oncology. And then third was Hercules CM, which is obesity. So again, looking at varied indications, right? But to your point here as the tease is that immune-related indications were 4 of the top 10, which really demonstrates that the industry is moving in that direction and seeing that as an unmet need.
Right. And, anybody that's been watching the drug industry would have predicted some type of obesity related disease would get that. And anybody listening to technology would expect AI. But immunology and autoimmune is definitely a new entrant. Like for the past many, many years, we would expect this funding to be dominated by oncology companies and oncology startups because that's just where the highest number of new targets are. So definitely the impact of autoimmunity and immunology is being seen in the capital markets as well. So that was a big part of the story. The other part that I thought the story did a good job of highlighting too, though, is that the number of technologies that get funded is smaller, but the funding per technology is higher. And I think that could be for a couple of reasons. Definitely interest rates will reduce sort of overall appetite. But there's a recognition basically that you have to get farther along in the process now before you can move on to next steps, be that acquisition by a major pharma or actually selling the medication on a wide scale. So it just requires more capital to get to where you need to be in a steady state.
But that is also providing another benefit for these companies. Now, historically, these companies were seen as a little bit more risky, right, than public companies, for example, just because of the funding nature and all. But now that these larger mega rounds are happening, it's attracting C-suite people that may have generally gone to public companies. And so the improvement would be in, I guess, theoretically, better management, right? Or at least a broader skill set, perhaps, than you might have otherwise seen.
Yeah, no, you're absolutely right. I'm sure with that, higher level management and executive presence, we'll get a lot more sort of efficient performance of these systems as well, because that should mean that these people have been around, have seen things. Doesn't always mean that it's better, right? But in a lot of cases, they know how to make companies very efficient. So yeah, absolutely hopeful for that and looking forward to what this next year brings, which is, of course, why I put JP Morgan 2025 on here as well.
I think as I was going through this one, it struck me, I think that was one of our first podcasts we had mention of JP Morgan. I don't know, maybe it might have been our third podcast or something like that. But yeah, this is a bellwether type of conference. Some of the things that are driving the mood around, obviously, anytime you have a new presidency, there's some uncertainty as to what that means for healthcare here specifically and the changes. The Inflation Reduction Act it could spill over, reduce pricing. And so that's another thing. What is that going to mean for the big pharma players specifically? But then also there is the thought that there might be more M&A activity moving towards this year. And so that could be a benefit as well. What were some of your takeaways?
Yeah, I would agree that there's definitely a level of uncertainty around the new administration. I would call it cautious optimism at this point. So that's always a good thing. Mm-hmm. And yeah, the big takeaway for me in terms of M&A so far, because right now it is we're recording this as JP Morgan is going on, so we've seen a couple announcements come out. So, I think it sets a pretty good tone for 2025. So I'm pretty happy with that. But the other big thing that I saw repeatedly in a number of different articles was the impact of drug development in China, and what that means for the innovation ecosystem in the US as well.
Yeah, it's a big one, right? Because the cost there is somewhat lower than it is in the US, European companies. And so big pharma is certainly looking to bring that in and bolster their assets, right?
Yeah. So I know that'll be a closely watched parameter too. How many of these deals and M&A deals that are being done to license assets developed in the US or EU or China? And what does that mean moving forward?
So I do have two things on this. One was a note that stuck out, and I don't know if it stuck out like a sore thumb to you as well, was the mention of the upcoming expiry of Keytruda. So the patent exclusivity there. What are your thoughts
Yes.
about?
No, and that's actually sweeping the industry. A number of different companies are facing patent cliffs. So they are working to deepen their pipelines and broaden their pipelines now. That way they can be prepared. This happened previously in roughly 2008. And a lot of people think that the market did not, or the major players in the market did not respond quickly enough to the patent cliff that occurred then. And then that led to them paying too much money for a lot of the assets that they had to bring in to shore up their revenue. So now you're seeing a lot of players start to go in early and ideally spend smaller amounts on acquisition to make a sort of prospective bets on what's going to be their biggest revenue providers moving forward.
Yeah, absolutely. And I mean, Keytruda has been a core in therapy, for quite some time. You get patent exclusivity. It's one of those things where a company has the window to be the only seller of that therapy, to be able to recoup development costs and whatnot before it goes to generic, or in this case, it would be biosimilars. And so that was the other thing that I kind of wanted to and talk to you about was biosimilars, right? And how this is something that, yes, it's supposed to be efficacious in the same manner, safe in the same manner, but it's not exact, because what you're talking about is using cell lines to produce an antibody, and there could be differences such as glycosylation or protein folding, and so there definitely is the burden on demonstrating that it's similar in that way, but certainly not exact like you would have with generics.
That's exactly right. And so the patent cliff may not be as severe in a lot of cases with biologics, because these are definitely much more difficult molecules to produce in the first place. But then, as you say, much more difficult models to produce consistently and then demonstrate the same level of efficacy there. So I think that generic companies will have more of an uphill battle in terms of doing that. I know some have already been done, but for things like Keytruda and other extremely effective antibodies, it may be quite difficult. So absolutely correct there.
Mm-hmm. All right. So what do we got next?
Next up is moving on from investments to KCAS news. And in terms of KCAS news, in keeping with the investment news, that was really a look back at 24 and look forward at what we think JP Morgan means for 2025. For KCAS, we just had the entire executive team basically give their thoughts on what's going to happen in the bioanalytical industry from what happened in 2024 and what's going to happen in 2025. So a lot of interesting takes on different parts of the industry and how it's going to affect different geographies there.
Now, how did you like doing that compared to doing a podcast?
I mean, I had to do all of my own talking. It was terrible, Adam. So, from that standpoint, I find a conversation to be easier. But of course, we have a wonderful marketing team, so they made it as conversational as possible.
I'm curious though, was that scripted, or did you kind of you prepared and really just did it from notes on the fly kind of thing?
That is primarily on the fly, where I knew roughly what was going to be asked, and much like the podcast, we've got a couple bullet points, and that's it. So
Yeah. No, it's cool to know. It's always good to get perspectives like that, and also look back at what was accomplished. I mean, did we do anything last year? I forget.
I remember playing ping pong quite a bit, but that was about it too, Adam. Yeah, it's
Was it in Australia or was it in Lyon? Actually, I think it was in both. It was in Switzerland too. You were playing ping pong all over the place last year.
yeah, with airplanes, I guess, instead of paddles, right? So
Yeah. Lots of stuff going on. So it was a very exciting and fast-paced year last year, but certainly 2025 shaping up to be even more so.
yeah, absolutely. And so that brings us into 2025. And one of the things that we think is going to continue is this ongoing investment and focus on autoimmunity and immunology related applications, and especially in flow cytometry. So we do have a paper, and maybe now is a good time to introduce our two guests in more detail as well. So Allie and Ayman, if you wanted to introduce yourself and a little bit of your background.
Sure. Thanks for having us on the podcast today. My name is Allie Greenplate. I am the Director of Strategic Alliance for the Institute for Immunology and Immune Health at the University of Pennsylvania. A little bit of a mouthful. I am a PhD by training. I did a PhD in microbiology and immunology at Vanderbilt, where I looked at systems immune monitoring in patients with cancer as they undergo treatment. I came to Penn in 2019 to do a postdoc with John Wary, and the rest is history. I've moved into this staff position with Immune Health, where we're looking for ways to monitor a person's immune system and to decode that as a way to tell us something about their health trajectories and outcomes.
Hi, my name is Ayman Rezk. I'm the flow cytometry lead at the Immune Health Initiative at UPenn. Prior to joining UPenn, joining Immune Health, I've completed a PhD and a fellowship in the field of neuroimmunology, where I focused on the cellular immunology of multiple sclerosis, specifically trying to understand the antibody-independent function of B cells and how these may contribute to the dysregulated immune responses seen in those patients. Along the same lines, I've also investigated the changes observed following treatment initiation and trying to identify immune signatures that can predict treatment responsiveness. Since joining Immune Health, my goal, along with the wonderful team there, is to apply the high-dimensional cytometry tools to tackle other diseases at a larger scale, and as Ali mentioned, to be able to develop immune tests that could reflect the health of the individuals, and for it to be scalable for clinical for the integration to the clinic to improve patient care.
Okay, excellent. Thank you. And as part of this discussion, we will have a paper that will sort of bounce in and out of. So this paper is Sequential Immunotherapy Towards Cures for Autoimmunity. It's written by Francisco Ramirez Valla and his team at Bristol Myers Squibb. So that's just two co-authors here. So Joseph Marinville and Sophie and Robert Plenge. Sorry, three co-authors. Yeah.
And it's in Nature Reviews. So Nature has an impact factor of 50.5 as of 2023. So very high up there. I think anybody in science knows Nature, right? And so it's one of those publications you'd love to get into. Has a lot of great information. And so going in, sequential immunotherapy, what are we talking about? Essentially three steps the authors lay out. Number one is controlling inflammation, and this is where the preponderance of therapies are at this point in time. Two is resetting the immune system, and one of the tricks that they discuss is really getting to that immunological memory. So these memory cells are the ones that, if they are not reset, could present a relapse, right? And then finally, if you get past those, once you get past those first two, you then promote immunological homeostasis, and so making sure that you're maintained, your cells are healthy, moving forward. And so, getting back to our guests, certainly it really interested to hear your viewpoint on this paper. Your backgrounds are absolutely perfectly suited for it. And so I'd like to kick it over on something that really struck each of you.
I mean, I found that the proposed framework was kind of quite balanced and nuanced and logical in that it's a good framework for tackling the treatment of autoimmunity, and obviously a lot of the treatments we currently have hit on those different steps. Still, it kind of provides, or at least exposes, the challenges that the field faces for long-term remission, addressing patient heterogeneity, disease heterogeneity, which is still going to be the challenge for us as we move forward, and how we can tailor the treatment of different patients based on their clinical history, molecular profiling, or the use of cytometry to gauge their immune health. I think that'll be the next frontier in the field of autoimmunity.
Yeah, I really like this framework and this way of thinking about it. I perhaps think of things less linearly in terms of time. I think there's a really interesting Petter Broden paper that came out, over a decade ago now, that looks at, the contributions of lived history versus genetic input and its influence on the shape of the immune system. And what they see is that 30% of the risk factor for autoimmunity comes from genetics, which means that 70% doesn't. And so as we think about the immune system and the different parts of it here, you have to start thinking about, okay, there's the underlying genetic predisposition, but then there's the lived history of a person as it looks in their immune cells. There's the interplay of their environment that can be read out in, proteomics in the serum. And so thinking about how to address each of those features, suppressing the inflammation and then resetting that immune memory is really interesting. I suspect that it will be less that every person will go through step 1 and then step 2 and step 3, and more so to Ayman's point that we need to understand where the majority of the, let's say, trauma is happening in each of the person. Is it that there's a specific immune cell subset, or is it that there's this history that's shaped the immune system into an autoreactive set? So yeah, I think it's I like thinking of it in bins. It at least helps my human brain parse the information much more easily.
Yeah, I think you bring up a great point about that bias between whether it's genetic or is it environment. One of the things that they mentioned near the end was molecular profiling and really looking at the relationship between the molecular features and the clinical outcomes, which I think is going to be very informative in really elucidating what is going on, because it is so diverse, and that's where the challenge is. There's a lot of information out there. The next step is really trying to pull that all in there, and then you can tailor it. Like you said, it may not necessarily be all 3 steps in that same exact order, but really understanding what is driving it. Yeah, it's very interesting. I think the last part I have about that molecular profiling was just that they did note that, for example, DNA methylation changes can be reactive to the disease and not necessarily the cause. And so with this sort of information, you also have to be cautious about understanding whether it is a reactive or causative agent there.
Yeah, it's interesting to think, is there an underlying inflammation status of a patient that pushes them towards this inflammatory state? Or is it the inflammation of a specific organ that then disseminates to the rest of the patient and causes some of these changes? And I don't think we know for many of the autoimmune diseases. My suspicion is that we are kind of on a fact-finding mission right now. In the early 90s, we hadn't sequenced the human genome, and so we couldn't really ask these specific questions. We just had to collect the information first, and then ask those questions. I think that's where we are with immunology and especially autoimmunity right now. Like, we need to do large-scale information collection, and then start to ask these questions.
Yeah, and immunology, it's a challenge because the paradigm for a long time in therapy was one gene, one therapy, right?
Yeah.
And that's just not the case here. I mean, in some aspects of it, but by and large, there's a lot of interplay, various aspects.
And interesting too, Allie, that you said, my human brain can only comprehend these things, or that's how we sort of make a story or put together a narrative for it. But you're absolutely right that this is a complex and dynamic environment with a lot of different factors sort of pushing us one way or another. So, that's definitely an area to explore. And as you say, we need to have something like the Human Genome Project, but for immunology, to be able to understand that on a complete basis. Now that we have high dimensional flow that can go up to 40 colors, do you think that we have that at this point, or do you think that we are moving in that direction?
I think we're certainly moving in that direction. I'll let Ayman talk about flow cytometry specifically, but I'll kind of get the lead up and say that I think we have the technology now. It's certainly high dimensional and high throughput. The challenge, I think, is that, if you look at Flow Repository or any of the other repositories, there's a lot of data out there. It's not for the lack of data, it's for the lack of annotated data. As we think about scaling to the level of the Human Genome Project, for example, you really need to be able to tie an immune profile to clinical features, reliable clinical features of a person. And so I think that's where we are now. We have the technology, but we need to be really thoughtful and organized about how we collect that data, and of course how we design those panels in a way that doesn't produce artifacts.
So totally agree. There is recent progress in this arena where high dimensional flow cytometry has helped identify subgroup of patients that may have different clinical presentations or clinical or disease activity, and or can also predict who would do well on certain treatments. So I think that's where the field is. As Ali mentioned, the challenge has been that the nomenclature we use in the field of immunology still a bit needs to be agreed upon, so we can all share files and extract more information, and with that integrating the clinical metadata, other factors that may influence the heterogeneity in the responses based on environmental factors and such. And there is also still early progress done in charting the immune response in different diseases over its course. So whether we can profile individuals who are at risk of developing a certain disease in early stages of the disease, and as disease progresses, I think at different stages we're going to find out that there are going to be different immune signatures that are probably require different therapeutic intervention. I think that's where where we need to spend more, at least put in more efforts to be able to tailor the medicines that we give to our patients.
That's exactly it, right? And there needs to be standardization of the wet lab. What assay are we actually running? Like what panels and what antibodies and what fluorophores. But then, as you say, a lot of the challenge is actually the documentation piece. So, the way I think about it is a three-prong approach. So, at KCAS, we have these pre-validated panels that we have developed for autoimmunity. That way we can say to sponsors, run these across all of your trials, and they will give you a good readout of what we think are the best markers for autoimmunity. And then with groups like SolCap and CDISC, we can point them towards sort of unified naming of what these populations are. That way we get away from, is this an activated cell or a memory cell or something like that can mean different things to different people, and go towards a very scientific annotation, much like the Human Genome Project of this is CD63 positive or CD47 negative or whatever it might be, and basically make something that is machine readable. But you're right that it does miss that back end of how do we tie this all together with the patient data as well to make it fully translational. And that becomes very difficult because a lot of that is locked up in proprietary clinical systems so that one company could have a lot of it, but it's difficult to move the whole field forward at the same time.
Yeah, I think that's where something like an academic institution like Penn really fills that gap. We can talk a little bit more if it's of interest about how we, validate our panels and have our annotation. But because we're within a health system and because we're a research organization, we do have access to that clinical metadata. And so we're able to do, fully from your EHR to your immune profile to annotated information to a storage and sharing database. We're able to do that, front to back, which is really nice. And I think it's something that's not always possible, like you said, with pharma and other companies needing to protect that information for business reasons.
Right. There's just so much information out there. It'd be important to be able to tap into that and figure out a way that everybody benefits from that sort of information and the sharing of that information.
Absolutely. And, I guess the other thing that always strikes me about these treatments and this treatment landscape as well is exactly as we spoke about with Sana, where they were originally developing treatments for oncology, and now they have moved into autoimmunity and immunology related diseases. What role do you think sort of all of our learnings around oncology have played in how we think about these things, how we structure these boxes, and the status that we are with developing treatments?
So it's interesting because a few of the successful therapies used in autoimmunity were first developed for blood cancers. So anti-20 rituximab was first developed for B-cell malignancies and BTK inhibitors as well. And the most recent ones are the CAR T cells. So I think for some indications where we know that there's a shared immune cell populations involved, that's I think helped a lot to kind of accelerate the repurposing of those drugs. And I think I came across recent articles suggesting that even there is movement towards using checkpoint agonists for controlling or at least treating autoimmunity, which is kind of the inverse of what's been done in the field of oncology, where they use obviously the checkpoint inhibitors to control the immune response. I think we've learned at least quite a bit from that field, and probably has accelerated the integration of those treatments into clinical trials.
Yeah, you brought up a great point. I mean, it's amazing how certain targets have become so ubiquitous. And obviously, it's because of the cell type that they are affiliated with, right? You mentioned about CD20 rituximab, CD19 in CARs. I mean, it's been so broadly applicable both for the B-cell lymphomas as well as for the autoimmune space. And then, obviously, with bispecific antibodies, we have CD3 redirection. And so just really, again, being very smart about the design and targeting here it has paid dividends on multiple areas.
That's it. We've been so focused on, immunotherapies and even like the microenvironment around tumors that we've discovered a lot about sort of disease state biology, and then the ability to use that same language or those same pathways to then look into autoimmunity has been quite helpful, I think. And I put on here as a last bullet point about the frontiers of autoimmune research in both the research and development spheres. And with that, I was really thinking about sort of the academic and pharma environment as sort of early discovery and research, and then CROs and clinical trials as sort of ongoing development, if you will. So I think we've done a decent job about talking about some of the frontiers, but Ayman and Allie, any other thoughts or different directions that you wanted to cover just with the frontiers of autoimmune research on that early space?
But I do think there is some exciting work happening in the field of resetting the immune systems in patients with autoimmunity. We haven't covered that, but there was earlier trial done in Canada many years ago now, where patients with MS who had very active disease were essentially enrolled into a trial where their whole immune system was wiped out by using a combination of chemotherapy, and then they were reinfused with their autologous stem cells. What's interesting about this trial is that the patients who went on to not have any disease activity for up to 15 years in the absence of additional treatments. And I think that's going to be, it's an example where if we can find a mechanism to, reset the immune response in those, high risk individual or individual who have high disease activity, there may be long-term remission in that regard. And just to plug in that, I think that there is an ongoing clinical trial called Beat MS taking part in the US right now, where they're aiming to kind of compare side by side autologous stem cell transplantation with the best available treatment in patients with multiple sclerosis. So I think that's going to be an exciting, trial to follow and to see how it plays out in terms of safety and efficacy.
Yeah, I think we're also, I think we're entering the age of scaling immunology. We've talked a little bit about the Human Genome Project, but we can see, I think, glimpses of this now with the UK Biobank releasing O-link proteomics for 600,000 subjects with tied to EHRS data, or EHS data, sorry. I think that one of the big outstanding questions, so I think Ayman did a really great job of drawing our attention to the clinical breakthroughs that I think are about to happen. And I think that there's one of, to me, the biggest underlying questions is what contributes to the onset and the continuation of symptoms in autoimmunity. And I think this is where the large-scale immunology with biobanks and concerted scaled efforts is going to be really helpful to help pick apart the heterogeneity in genetics and lifestyle of subjects. So I think the next thing that we're going to start seeing on the more basic side is just simply scaling the collection of data in the immune space and applying that along with machine learning and AI to help pick apart these really complex systems.
That's it, right? And that scaling piece, I always view as part of the development efforts, where you get an assay in. Is it robust enough to run in multiple locations and on multiple instruments and with multiple people that you would need to do this? But then can it be miniaturized? Like we're talking about reagents here, and for our general viewers, I'm sure will be familiar, but an individual antibody fluorescently tagged can easily cost upwards of $300 for a lot of them. So you talk about then needing to do 40 color panels, and you're talking about needing to stock over $12,000 worth of reagents. Now, you don't use all of that on a single sample, but even if you use a small bit of it, $12,000 is pretty hefty for a startup fee.
Yeah. I mean, I think when we cost out our assays, 75% of the cost is just antibodies, and that includes people's time. It's an expensive field right now, but I do think, like the Human Genome Project, that the technology will develop as we push it. And so focusing on that miniaturization and scalability, I think, will revolutionize the way we think about these types of assays.
Right. And as you say, it's definitely information collecting now, but we can see a future where we can have a subset of assays or a smaller set of assays that could be used for screening tests to then direct you to the right therapy or to better specify. So instead of just general lupus, is it subset A, B, or C, and things like that. And the same with multiple sclerosis and other types of autoimmune disorders there.
Yeah.
Yeah, I mean, it's really going to come down to understanding. I think one of the things that this paper that we talked about had me most interested in was making it's more smarter weapons, right? Against autoimmune diseases and specifically against memory subsets. Right now, we can attack very broadly on CD20, CD19, but then understanding that what does that leave the patient with afterwards, whereas if we can even more finely tune these things against memory B cells for specific antigens, then all of a sudden we change the game. Same thing with the T cells, who really just exactly targeting what we want and leaving everything else intact.
Yeah, and I think I got thinking. I mean, this is a really great paper, and you can't put everything in a paper. We have limited space, but it got me thinking a lot about not just the B and the T cell memory, but the innate cell memory, like the epigenetic state of those cells having existed in an inflamed context for so long. I think we start with the B and the T and the plasma cells and all of that, but as we go to then step 3 of I forget what they called it, resetting. No, that was step 2. Anyway, we go to step 3. How do we actually heal the memory of not just the adaptive, but the innate immune system?
Okay. Definitely an exciting time, both with flow cytometry and also with autoimmunity. So I'm looking forward to a number of the other things that we're doing as part of this effort in Q1, Q2, but also more conversations with Allie and the team at UPenn around that. But I did want to move us on into some of the closing segments of the podcast, just to respect everyone's time. In terms of fan feedback request, the request for this time is, what is your favorite or least favorite type of disease state indication to work with in flow cytometry? And Adam, correct me if I'm wrong, I don't think that we've asked this before. Is that right?
No, I don't think.
Okay.
And it's the way that you phrased it, favorite. I might have went most interesting.
Most
but
interesting. Fair. Yeah.
I certainly get what you were going for.
Sure. And source of that question is, in terms of working with lupus cells or working with AML cells or working with, just healthy donors, anything that you've found particularly challenging or particularly rewarding in terms of how to deal with that. So hopefully our listeners will give us some feedback there. And then, also just to wrap up, now that we're in January, it's a little bit farther into the year, but I did want to ask about any interesting New Year traditions. I've got quite a few colleagues from China, and I've found a lot of their Asian New Year traditions very interesting in terms of, always wearing new clothes and things like that. So wanted to hear from the team here on the podcast. What do you do for the new year?
I go to bed at 8 p.m. It's so exciting.
Excellent. Ayman, what about you?
Well, my spouse is Greek, so there's a whole lot of traditions there. But maybe one of the most fun one is that they typically bake a cake in which they put a coin, and the whoever finds that coin will have a good year ahead of him or her. So it's one of those exciting things where as the cake is getting cut, everyone's like, "Oh, is it me? Is it me?" So as things like that, people I think are fun to do sometimes.
Excellent. Nice. Adam, what about yourself?
So you'll have to forgive me. I have probably a two-part response, and hopefully we don't get down too far of a rabbit hole here. But the first part is just around, I think right now I'm kind of resetting my New Year's traditions. Usually used to do just had like old college roommates and hang out and whatnot. But having lost my son a couple of years ago, it had been one of those things that's really difficult to celebrate a new year because it's kind of like going into another one without him, right? But have been talking with friends, family, and everything. I would say the second part of it is my family tradition, the broader family. So we don't necessarily get the chance to get together on New Year's. And so we get together a couple weeks later, and we'd just done this past weekend, and my parents will actually tape the ball dropping. I say tape, record, whatever you want to call it on the DVR. They DVR it. There we go. So you know how I'm pretty old, and I did just get 50 plus vitamins. I told you that, Brian, despite the fact that I'm not quite 50, I'm almost there. Anyways, what we do is the ball drop, everybody counts it down, even though it's bright sunny out on a Sunday at like 2:00 in the afternoon. And then we do like some sort of little exchange where my mother requires that we each give her $20, and then she gives us like this sock with a bunch of stuff in it. And you are guaranteed to get your money back at least, but there's possibility for greater prizes, and you can like steal somebody else's sock, but you don't know what's in it. And so this time, my daughter didn't steal my sock, right? And when we opened it up, I had more money in my sock than she did in hers. Just like, that's just not right. I did make it right. I was like, here you go. I'll give you the difference here so that we both come away happy. But yeah, so again, long-winded. That's kind of par for the course with me. But how about you, Brian?
I love it. No, that's great. It's like Boxing Day where it's a little bit afterwards, and the white elephant or the sort of round robin gift exchange too. So that's wonderful. No, I am also very similar to Allie. I've got two little ones, and I've gotten very used to watching the Sydney fireworks display. So we'll put that on YouTube. Adam, if you want to move from VHS to DVR to this internet thing, then there's that YouTube available. So we'll just play the video whenever we want them to go to bed, and then they think it's the new year, and off we go.
What if I was on Betamax? Come on.
I mean, it is a better platform, so I understand why. But
yes.
Fair. All right, and then the last one, of course, very relevant to Philadelphia recently, is largest snow structure ever created.
This is a personal question?
This is a personal question. So, Adam, what is the biggest snowman or snow fort or snow thing you've ever made?
I guess if we're just talking height-wise, I made a snowman probably about 15 years ago that was about my height, so about 6'3". And then just making fort structures that cover, 20 feet by 10 feet, something like that, underneath the pine tree. So it was a really cool thing, but nobody attacked us. I mean, that's a bummer. You build it so big, and then there's nobody there to get at you. So, yeah. How about everybody else?
I think I'm going to count this as mine, even though I was a child. But in the Philadelphia snowstorm of '96, my dad, who's an engineer by training, built a gigantic life-size igloo that me and my two sisters, I'm going to say helped with, but I'm sure it got in the way. But we were able to like hide in and do all of that. So in my mind, in my memory, this was gigantic. It was huge. It was probably 5 feet tall.
That's really cool. I like that.
But it's a good memory.
Mm-hmm.
Very nice. Ayman, what about yourself?
I'm sad to report that I've never had a chance to build one, so now I have to wait for the next winter to get on this task.
You were in Montreal. What were you doing?
hiding inside. It's really cold. It's not,
All right.
to stay.
No, I feel you. Adam is very familiar with my allergy to snow, and yes, indeed, is not to be toyed with, right?
Yes.
He is absolutely 100% true. I mean, he hates the snow. It's pretty funny. The second, like yesterday, it started flurrying out, and like I couldn't even see the flakes. He goes, "Oh, what are we gonna do?" I said, "What?" He goes, "It's flurrying." I was like, "Oh yeah, I guess it is." Okay.
Here's my proposal, Ayman. The next time it snows, I think we get KCAS involved. I think we build a snow germinal center. Like,
there we go.
You know,
there you go.
Our next big thing, our next big project.
All right.
Add it to the list. Wonderful. All right. Excellent. Well, it has been absolutely fantastic, Allie and Ayman, to have you on, and loved our conversation. So thank you, and thank you to all of our listeners. Our next recording will be in February for episode 17. We're targeting biggest predictions for cell therapy in 2025, and we'll see how that shapes out.
Yeah, Allie, Ayman, really a pleasure talking with you both, and wish you both the best, and thank you all to our listeners.
Thanks for having us.